Okay. Welcome everybody. It's my great pleasure to be back to the early spring quarter series of the VMOL seminar. And like this quarter, we're gonna focus fully on GNPS2. And what else than to kick it off with like the creator himself. So it's my great pleasure to have Ming Wang today give the first seminar of this series. And just to give you some brief background, Ming did his undergrad in computer science at the University of Illinois before he then went to UC San Diego to do a PhD with Nunu Bandera and Peter Dorstein, where I had the pleasure to meet Ming back then when I was a grad student as well. And since then we've been co-workers there later on as postdoctoral researchers. And now, like a little bit more than a year ago, for 2 years now, Ming joined UC Riverside here as an assistant professor. And I don't know, so somehow I also had the pleasure now to join Ming here, and we're direct neighbors. So he's focusing on computational metabolomics, and I guess most of you know him for like being the main engineer behind GNPS1 and now also GNPS2. And without further ado, let's just mention very briefly one technical aspect. There will be a hands-on component today. And in order to follow it, if you want to, you will need a GNPS2 account. And Ming was so kind and provided a set of GNPS2 account tokens for us. And in a second, I will paste a spreadsheet link in the chat where you can put in your name and affiliation and eventually email address. And then we can provide you a token there in case you want to sign up for one and follow the hands-on examples. In case you don't want to follow right now, that's also fine. I think you can just ping Ming later on to get a token, and then hopefully this will be done. Okay.
Yeah, you actually don't need a token to follow along on today's kind of interactive examples, but some of it, hopefully you want to use it anyway. So, okay,
Cool.
we'll get started. We'll get
Without further ado, floor is yours. Looking forward to the seminar.
Okay. Well, thank you, Daniel, for the wonderful introduction. Yeah, it's well, I'll ramble a little bit, but in the chat, Danny will organize some of the tokens. But as Daniel said, I'm an assistant professor over here in the computer science department at UC Riverside, and we're developing some new tools. But one of the things that we found, or I personally found pretty rewarding as a scientist, is not only do you want to make technical innovations, right? In my field, computer science, but you also want to you want it to be easy to use and impactful, right? So it's not sufficient to have a fancy paper. Not that we publish that many fancy papers, but you can imagine you have a fancy paper out. But then if it doesn't help anybody kind of accomplish their goals, then kind of, so partially what's the point, right? So especially this is, in chemistry, it's a little different, but in computer science, especially bioinformatics, this is such an important aspect. Our whole goal is both to advance the field, and in the name, it's bioinformatics. You have to make some sort of discovery or help people make some sort of discovery. So impact and usability are keys. And so GNPS2 is kind of a piece of this puzzle. It's, a graphical user interface with no substance is not useful either. You need to both do the computational advances as well as have a vehicle to deliver those to a wider audience. So throughout this kind of seminar series, we're going to talk both about the user interface, starting here in terms of GNPS2 to help make things easy. But later on, we're going to talk about some newer tools and computational advances that we think are the substantive part that meets the kind of the easy to use portion. So that's just kind of setting up some of the stuff that we'll talk about. But okay, so without further ado, we'll get into this. And so as Danny mentioned, this all started at kind of UCSD when I was a grad student as kind of a side project that was kind of fun to play with. And it grew into something way more way more popular than we anticipated. We were just very lucky that that the community was willing to even use our tools. That is, some of it's hard work, but it's mostly luck. So we were just very thankful of that. But the way I see kind of the pieces of GNPS2 and just in general, kind of the community and the computational bits, there's kind of three big pillars that we would like to think about. First is the algorithms in the top left corner, the algorithms and visualization of some data, usually private data. In the top right corner here, the big data portions, right? So this is how can you leverage public data? And then there's this knowledge bases here on the bottom side. We're going to, again, throughout this series, we're going to talk about some advances in each of these pillars. But today is just kind of an introduction, going to get you all into the ecosystem. And so one of the things that evolved from GNPS1 at UCSD is that's going to definitely keep living on for as long as it can continue living on. But we want to kind of make things a little bit nicer and more modern. And one of the things that we've launched is GNPS2 at UC Riverside. And so this moves, or at least this creates some new capacity for algorithms and visualization and reanalysis at UC Riverside, whereas the big data repository stuff and the knowledge bases will partially remain at UC San Diego, and we still maintain some of those. And so that's kind of the relationship. So you'll see a lot of websites floating around, but this is kind of one area that one architectural design. And so just to give you a brief outline, there's a lot of visualization tools everywhere, but some of them are like, when we'll talk about these later on, the GNPS dashboard. Oh, so some of these are from some other slides, but the GNPS dashboard, which is a visualization of raw mass spec data, the metabolomics resolver, which is a way to start sharing MS1 and MS2 visualizations, especially for publication. And we also have interactive network exploration. So that's one of the things that we're going to look at today. And so some of the analysis tools that we're going to talk about later down the line, and some of it today, is molecular networking. So that's one of the more kind of a legacy, not a legacy tool, a classic tool. But we'll talk about that today and kind of the things and throughout this, things that we have enhanced with it. Another one is called the MASS, so the mass spectrometry search tool. How can we find and mine a huge amount of public data? So we're talking about billions and billions of tandem mass spectra. How do we start making use of that in the context of your own data? And finally, I guess not finally, one of the other ones is the mass spec query language. And so how can you start looking for exactly what you want to find? So this is what this query tool, MassQL, is useful for. And so I was wanting to put some of that in here. We did put a little bit of in there, but we've got to save it we can't stuff too much in these early slides. But cool. So that's just a high-level overview. Let me swap to another PowerPoint presentation really quickly. So here we go. So we won't go through too much of some of the really, really basics of logging in, creating an account. Well, I'll show you creating an account, and uploading files. Those are pretty self-explanatory. We have other workshops for that, and these slides are available for you all to look through as well. And so it's not particularly unique compared to GNPS1, but this particular slide deck, how do you access GNPS2? How do you create an account? How do you log in? So we'll just introduce you the very basics. So to very first go to GNPS2, go to gnps2.org. You can Google it as well. It's findable on Google now. And you'll see some page like this. Again, it's reminiscent partially of GNPS1, but it's a little bit cleaner. And if you want to make an account, you'll want to go in the top right-hand corner, and we're kind of slowly growing the user base. We are we have a very modest computational setup, so we want to grow modestly. We don't want to have this big explosion, then we run out of storage space and compute. So One of the things, so what you enter your username, password, email, and one thing that you'll need is an invite token. So this is one way that we're controlling this growth kind of very slowly. And Daniel should have sent around a link, so you can sign up for these. You can grab a token and you can put it into the website to sign up. So we'll give you a few minutes to do that. And Daniel, you'll let me know if we need more tokens, because the number of attendees here is a little bigger than we expected. So
I think so far we're good. I also have some extra ones, but I might ping you in case we run out.
okay, we'll let everything go through here. Give you guys a few seconds. Well, anyway, thank you all for showing up. Yeah, we're always very thankful when anybody shows up. It's you know, it's kind of like when you throw a party and you're afraid nobody comes. So, but throughout all this whole process, I think one of the things that really made the platforms much better and more usable over the years is we've learned a lot from what people actually want from the community. So feel free to chime in. We hold a weekly office hours on this quarter on Wednesday mornings, Pacific time. You don't have to make an appointment, or you should sign up. There's a little Google form, you sign up, but you do not have to contact me. Just show up, we'll be there, we'll answer questions, we'll get out. So this saves me responding to a million emails, so it saves my sanity, but it's also a lot nicer sometimes just to like have a conversation with somebody. So that's something that definitely consider taking advantage of. You'll probably get a more detailed answer there from the GNPS2 team versus an email. So, but I see there's a lot of action still happening in the forums here, or the Google Sheets.
Yeah, don't worry, Ming. I can monitor.
Okay.
I think I have another 30 or so it should be fine.
Perfect. Okay, so once you guys sign up, you'll be able to remember your password and you'll be able to log in. You can go ahead and if you forget or if you want to change your password, you can. But I highly encourage everybody to use a password manager so you do not forget your password. Again, this is this is my unsolicited advice for everybody in the long term. Okay, just because there's no password reset mechanism right now. Okay, you'll be able to log in, and then just very briefly, you can click the file browser in the top right-hand corner. You can make folders on the GNPS2 servers, and you can do drag and drop all in the browser. You don't need any external software. It's relatively simple to get data files over. Cool. Any kind of concerns or questions? Okay, so I saw a question. If there's an invalid token, make sure there's no trailing or leading white space. And if somebody else used that token, it will be no longer valid. So it's a one-time use token. Okay. So that's kind of the very high-level details. So one of the things that we want to chat about here is we'll just give you a very high-level overview of molecular networking for those of you who have never seen it before. For those of you who have, just kind of ignore it. But so kind of one of the main motivations is, especially in untargeted metabolomic small molecule data, you have a lot of spectra. You have a lot of tandem mass spectra that come out, right? So hundreds to thousands, even hundreds of thousands if you have a pretty large experiment. And so kind of sifting through all that is pretty annoying. So one of the key advantages of using molecular networking is it gives you a way to start organizing your tandem mass spectra in a somewhat logical manner so that you can prioritize what's interesting for discovery, especially if you want to discover new natural products or new metabolism products and things like that. And so one of the things that we can do is we can do library matching. So you match it to a reference library of known compounds to identify these things. But unfortunately, libraries are not complete. And so most of the data becomes, they're not annotated. So you want to, again, we want to start organizing these things. And so one of the ideas here in terms of organization is the intuitive organization is that imagine you did have two molecules where you knew the structure, and you put both of those molecules into the mass spectrometer, and they're very similar. So in this example here, again, it's a natural product, and it changes just the lipid tail. It's a very small modification. And if there's a small modification change in the structure, how does that manifest in the tandem aspect? And so broadly, the tandem aspect is similar, but not the same, but it is still very similar. And so one of the ideas is we want to be able to start capturing this similarity in the tandem mass spectra so we can infer backwards that they might be putatively structurally related. And so just giving you a small toy example here, we have two tandem mass spectra on the left-hand side and two molecules. Let's say that these are their true molecules structures on the right-hand side. And so what you can see on the top, we have a molecule, and on the bottom, we have a molecule that differs by some sort of delta. And so what happens is whenever you fragment both of these molecules, what can happen is the fragments can either have the same mass, or it can either be the exact same substructure, or it can have this delta modification. And if it's the exact same substructure, that means it will have the same mass, right? So you can see these vertical lines here. They'll have the same mass because of the exact same substructure, exact same fragment. However, if the fragment includes that delta, it will have a different mass. And the different mass will simply be it'll be the same peak at the top, except it's shifted by this delta. And so that's where you can see some of these cases where it's shifted. And then this is kind of the bottom line intuition for the alignment idea here. And so given that this has, somewhat happens in real life, we can align these two spectra by saying it can either be at the same mass or it can be shifted by this delta. And then we can consider all possible scenarios and this can happen. All the peaks are the same mass, one peak is shifted, another peak is shifted, or both peaks are shifted in this particular toy example. But at the end of the day, we want to consider all possible of these alignments between these two tandem mass spectra to be able to start capturing potentially these modifications. And so if we do that, we are now able to kind of putatively say the similarity between two spectra. We can start scaling this effect up and start doing all pairwise similarity between your entire data set. So we can ask, hey, every spectrum, how similar are they given this sort of scoring and alignment scheme? And so at that point, what we can start doing is we can start organizing these into molecular networks, where each circle, which node here represents a unique molecule in your data, and these lines represent significantly high or a high similarity given this alignment scoring scheme. And so we can start visualizing these things as these big networks. We can start layering on more information. For example, if we identified a particular molecule or node in your network as having a library match, that's a starting point for annotating entire families of these molecules and start propagating these annotations. You can start layering on differential expression information and things like that. And just as a visualization, here is one particular molecular network that we published, I think, a couple of years ago of some natural products, where you can have distinct families of different types of natural products that kind of self-assemble. And again, you can see that we started off from looking at pairs of spectra, and now you can start doing this at hundreds to thousands. You get these large networks that you can start exploring and prioritizing. So that's kind of where we live, or kind of the intuition of this molecular networking idea. And there's many different variants and flavors that have grown over the years, and we'll definitely start exploring some of that as this seminar series goes along as well. So any questions so far? Feel free to chime in on the Zoom or in the chat. I think hopefully all of you were able to sign up for an account.
Does this, I have the one question.
Yeah, go ahead.
Yeah, this applied for the plant metabolomics also, or is this is only for microbial
Right.
network?
So it's been applied very broadly for plants already. So the key idea here is that you actually have some similar molecules. Now, if all of your metabolites are totally different and totally independent of each other, and they're not structurally similar at all, there's no metabolism happening, it's probably not that useful. But in biology, generally when you have biosynthesis, and it's probably especially in plants, you have huge amounts of dense structural diversity. And so it's pretty applicable in plants.
Because in the medicinal plant, they have very large diversity in the terms of the secondary metabolism. So that might be it cover all of them, or
Cover, what do you mean covers all of them?
it's not possible, no?
Cover what? What
The
covers
class
what?
of the metabolism for the medicinal plant, they have very large variation in the terms of flavonoid, isoflavonoid, and carotenoid, apocarotenoid. These are the very diversity in the medicinal plant they have. So these database can be fulfilled because these are the very diverse metabolite. They don't have the even the commercial standard. So
Right. So right here, we're talking about molecular networking, right?
yeah.
So molecular networking, as I explained, is a technique to start organizing related molecules together. It doesn't matter if you know what the molecule is. It's based on your own data.
Oh, thank you.
Yeah. Any other questions? Okay, cool. So let's get started on a hands-on version. Here we go. So a hands-on, let me full screen this. Here we go. So in this particular portion, we can talk about how to run stuff. We're not going to dawdle too much on it to have you recreate this just because we only have 1 hour to go through all this. But we'll provide these slides so you can actually look through it. But one of the techniques is that we just talked about is molecular networking. And so you can apply classical molecular networking where you come in with your raw, untargeted tandem mass spectrometry data in GNPS2 and just get to a molecular network without too much fuss. So just as a very high level, once you log in, you can see there's a launch workflows. There's a big table of workflows that you can kind of start searching through. There's an actual search box now. So you can find classical molecular networking, and you can start selecting data. Again, there's a little file browser to select data that you want to analyze. There's some public data that we can select. Again, this is just to warm you up to the idea. So this is not the first time you see it when you actually go through these slides. But at the end of the day, you'll get to some sort of task result here. And so let me actually post this in, here we go. Let me post this in the Slack or in the Zoom channel. And so this is, once you've analyzed some data, this is the result page. And so one of the key things that we'll want to start looking at is visualize full network in browser. So let me just do this actually live here. So kind of in difference from the things in GNPS1, you can visualize the full network in the browser here. And at least within reason, there's kind of a size limitation of a few thousand nodes. But there's a lot of interactivity and shareability that we can go through here. And so one of the very first things you'll notice on this left-hand side, you can, okay, so like my scroll is a little aggressive. So you can see there are circles here that represent unique molecules and connections between them that represent putative similarity in their structure. And so you can start scrolling and navigating around these networks. And so kind of the most basic things is you can start visualizing the tandem mass spec fragmentation. So kind of expected, not the most interesting thing. And also feel free to click on this and follow along and play around. Let me also send this particular link in the chat here. So one of the things that we can start doing is we can start changing the labels on these nodes. So you can see these are just numbers. They're not super informative. But let me just keep zooming in here. But what we can do here is if you look at these node labels, you can change this to several different things. You can do a summary. It tells you M over Z and retention time. So that might be helpful for some people. If you want to look at the library match, it'll give you the putative library match compound name. And so these are some amino acid something. But this family of different tyrosine, I think there's some lipid tail along with it. But this might be a way for you to kind of explore these networks to see if there's anything interesting they're identified. And then the other things you can do here on this right-hand side, in case this matters, you can start sizing these nodes by any number of different things. So you can do by M over Z. So again, this is probably not the most interesting, but you can see which ones are the big families. Sorry, my scroll is a little wonky here. So what the big molecules or the small molecules. Another thing, if you're more on the natural product side, you might care about things like mass defect between 0 and 1. It might give you some hints on unique elemental compositions. But that's one. These are one other things that you can start doing. Other things that you can start doing is, if we start zooming in here, is if you want to start visualizing spectra that are similar. So let me just randomly pick something around here. If you want to look at two molecules that are connected in a network and you want to ask how similar are they from a tandem mass spec perspective, you can start clicking on it. It'll show you both of the spectra, but we'll also start showing you the MSMS alignment here at the bottom. So you can start looking at what's the mass delta, what's the similarity score, and here is a starting point to start propagating annotations. So there's a later thing we're going to show you that makes it a lot fancier, but we'll get to that in a second. But another thing that we want to kind of highlight here is imagine that you know a lot about mass spectrometry, and for example, you want to say, look, I want to look for all molecules in my data set in this molecular network that have an MS2 fragment of 104.1. So I'm just highlighting that this is a random peak mass, because you know that means something about your fragmentation or your class of compounds, and you want to start looking for that. And so what you can start doing is there's this 104.1. So let's go down here. There's an MS2 M over Z highlight. So 104.1. And so I enter that here, and it looks for, across this entire data set, all MSMS spectra that have that particular peak. And we can see it's located predominantly in these two families of molecules. But this gives you a way to start prioritizing molecules to start looking for that has a particular fragment of interest. And you can be much fancier with this. You can say, okay, it has to have 104.1, and it can have certain ppm tolerance. Or you can say, well, it also needs to have a minimum intensity. And you can start pairing that with other M over Zs that have to occur in the same MSMS spectrum. So if you want to start doing, adding on more complexity in terms of what you're looking for, it naturally handles all of this. So there's a question in the chat: can you look for neutral losses? So yes, you can look for neutral losses from precursor as well as neutral losses between peaks. There's a way to specify this. And so this is wrapped up in the mass spectrometry query language, which is supported fully in here. So we'll talk about that a little bit later. And so how to be really, really powerful with that. But I just want to kind of tease the capability here where you can search for a single mass very, very easily. And then once you kind of play with that, you'll think about, as you were asking, Stefan, well, what if I want to be more complex, right? So this is kind of where we're starting and giving you the capability. But if you do want to be more complex, there's fields in here in order to give you a lot more like flexibility and power and looking for what you want to find. Any questions so far? So here's a question. If you want to see single nodes that do not network to anything, if you go back, there's another link down here called Visualize Full Network with Singletons. So if you click on that, what this will show you, hopefully this is the danger of doing stuff live. So it's loading. Okay, so what you can start seeing here is you can see all the families that connect to something, and there's a bunch of singletons down here. Now, one of the kind of powerful things that that you can do here is imagine that you actually want to start capturing more singletons without running these networks again and again. So you can start you can rerun these networks interactively here. And so this is like you can start changing these parameters. You can start changing the minimum score, and then you can click recompute network. And so what you can see here is it injects some more edges into the network, right? And we just did that live. And then you can start resetting. You can reset the layout. So let's reset. Okay. So sometimes it's a little wonky on the layout reset. You have to run it several times. There we go. And then so it reorganizes the network so that you can play around with these parameters kind of in real time and see if it helps you guys. So instead of running like this giant workflow that takes 15 minutes. But yeah. Does that any other questions so far? I think there were some questions here about NIST support. So one of the things with NIST and being a public institution is we will not publicly provide NIST because that's a violation of US copyright law. So we're not in the business of violating laws. So no, we are not supporting things that could possibly get us into trouble. But NIST definitely provides some nice software. Yeah, you can get a license from them. And I believe in the newest version, you can't even actually export it anymore. So you have to analyze it on your own computer. But one of the things you probably could do is you can download the exports from GNPS2 for your data, search it against NIST, and import those and overlay those IDs back on the network. So if that's something you're interested in doing, definitely give that a try.
Yeah, if we have a latest NIST library with our institute, that can be used in the GNPS.
No.
Oh,
it cannot. Because NIST, just first of all, we do not want to host it. And number two, I believe the newest version of NIST does not allow you to export the library, period.
okay.
That is something that they have stopped allowing users of NIST to do starting in the 2023 release.
Yeah. Because we have the latest 2022 NIST here. So I thought we can use in this.
Well, yeah, you cannot because you can't even export it. So
Yeah.
Any other questions? So here's a good question. So how do you annotating your own nodes? So there is a workflow. I don't know if it's linked here. So we need to streamline this a little bit better. But there is a workflow. Okay, there's a workflow called custom network overlay. So what you can do is you can input any TSV, CSV, or Excel file with your own custom annotations, your own p-values, or any statistical measure, any numerical or text fields. And it will overlay that onto the network. And then you can interactively explore it. So there's a workflow for doing that particular procedure. So there is a question about the Natural Product Atlas. What about the Natural Product Atlas? What's the question about it? Any follow-up about the NP Atlas? Well, so I just want to be clear. The Natural Product Atlas is a database of compound structures to the microbes of origin. That's what it does. It has no tandem mass spectrometry data. So if there is an identification in the public libraries that you can use a GNPS with structure, I believe we link out to the NP Atlas already in GNPS1, so you can kind of follow the links that way. And there's other resources where if we have structure and it's present in the NP Atlas, we link out there. Okay, so let's keep going. And so there's one final, a couple final things that I just wanted to show off really quickly for overlaying. So I'll include this in just a second. So one other thing that I didn't include in this example here, but we have this example right here. So let me include this for everybody. So if you want to overlay differential expression data to start prioritizing perhaps families that are unique to one experimental condition versus another. So imagine you have in this particular case, it's germ-free mouse versus a pathogen-specific mouse data set. And so you want to ask the questions, which metabolites are specific to germ-free mouse versus otherwise? And so what you can do here, there's a pie configuration. And so this is kind of the very commonly used things, is the pie configuration panel here. And so if you say yes, what it'll do is in group 1, colored in red, it'll show the germ-free mice, and in blue, the pathogen-specific mice. And so what you can see when we zoom in, so sorry about my mouse here. We zoom in, there are families of molecules that are blue only and red only. So we're going to kind of zoom in on the blue only here. And so what this will tell us is these molecules, this molecular family, is only present in, only present in the pathogen-specific mice, only in this group 2 condition. And so this might be interesting if you're looking for unique molecules in a particular cohort of data. And so one of the nice things is you can actually start changing these things around dynamically. And you can actually, we don't have this example here, but you can create meta groups. And so imagine you have 3 groups here. You can actually put both 2 of these attributes or 2 of these groups together and say, and then you can compare it to a third one. So this is not the most interesting way to doing it, but you could create meta groups so at analysis time without having to rerun and create new attributes that you would have to do in GNPS1. And so it just makes these analyses for comparing differential expression a little bit simpler. So again, you can start exploring these things. You can look at the pie charts, and that's some other things that you can do. Now, one of the other final things, imagine if you're in these networks, you change like the node labels, you change the sizing. Let's change it to M over Z. You change the sizing, and perhaps you want to recompute these networks to, let's say, 0.5 or whatever way that's interesting to you all. So let's see this rerun. Okay, so it's not significantly different than what we have right now. But let's say that, oh, there we go. And it just takes a little bit. So let's say that you rerun this layout. Maybe you've also highlighted, let's pick an MSMS peak of 839.5, right? So let's just put this here, 839.5. And so you've basically done some sort of analysis. Did that find it? 839.5. So okay, so maybe there's a little hiccup here. But we've done this sort of analysis and customization, and you want to save this or you want to share it with somebody. And the first person you want to share it with is yourself in 6 months or 3 months. You're going to do a favor to yourself. And so if you do if you click on this thing up here called link to this network, and copy link this URL. It's a huge URL, but it saves all the settings for this sort of analysis in a single URL, and it allows you to recreate it, and then you can give this to somebody else. So if you want to send it to your supervisor or a collaborator, and you've changed all these things, you simply send this giant URL, send it an email, and it'll recreate everything that you had changed in terms of settings here. And there are all these sort of customization things that you can do to make it even fancier. You can change these node highlight colors. You can change these high colors. Instead of being red, it can be, we can change the hue over here, and we can kind of see it changes the color here. This is not the most aesthetically pleasing, but these are sort of the customizations that you could do ultimately for publication if you choose, right? And so you can export to SVG, and you can pull that into Illustrator or whatever figure. And then we have some of these default, it's kind of nice one. These default styling, if you want to use our default styling. So these are different ways that you can start sharing these analyses and customized visualizations that might make your life a little bit easier. And so you can put these in your manuscripts as well. But yeah, so those are some of the unique aspects of GNPS2. And some of the things that we are pushing down the pipeline is we have GNPS2 obviously here running at UCR servers. And one thing that we're rolling out pretty soon, if you want to play around with the beta, we're slowly rolling this out, is we have a new website being hosted, GNPS2 in Germany. So there's going to be a redundant site in case we're doing maintenance in the United States. You can use it in Germany, or maybe you don't want your data leaving Germany. And we'll be able to synchronize your analyses between different sites so that your analyses will be available wherever we have this kind of federated system. So in case one site goes down or has maintenance issues, you can still analyze and access your old analyses, and it can be done in an automated, it was synchronized in an automated way. So creating some resiliency in the community. And so those are kind of the things that we'll be touching on. And one final thing that I just want to show you all that we're excited about here is we're covering a lot of ground just because this is the introduction slide. So there is a quick start GNPS portal now, a new quick start. It's called, everything's 2 now. So there's a quick start 2 for GNPS. And this is specifically for feature-based molecular networking. Again, we haven't covered this yet, but those of you familiar with it, you might be interested in using this to make your life a little bit faster, especially for collaborators that do not have a GNPS2 account. So you can run feature-based molecular networking, again, directly, doing the file upload and select the feature-based molecular networking, but something that, you don't want to do that. I'm pretty lazy. And if you go to quickstart2.gnps2.org, you're able to drag and drop your files, quickly select it, hit submit kind of in one go, and you can just submit quick start feature-based molecular networking. It's all done. You don't have to go multiple pages. It's a single page, and it just gets you really going very, very quickly in GNPS2 for feature-based molecular networking. And we're going to be enhancing this for classic molecular networking, plus there's like 30, 40 other workflows that we have on the system to make everybody's lives a little bit simpler. And finally, so that's one other thing. And finally, from a resilience angle, all the workflows that we have in GNPS to the extent possible so far, we've released it as open source. So you can run them, not with the old GNPS1, you couldn't run them locally. Now you can run them in Nextflow locally on your own Linux system. It has to be x86 Linux, but you can run those on your own platform. You have to be familiar with the command line. But If you don't want to use it on our system, you can run it on your own side. You can enhance it. You can change it. But anyway, so those are certain things that we've done in the system that makes it much more resilient and hopefully easier for the community to use. Just because the platforms and tooling has come a long way over the last 10 years. And so GNPS2 capitalizes on these things on the web side, as well as the workflow side, and just making it more accessible and more useful to a wider audience. So I know that was like kind of a high level, a lot of different things, but especially for those of you who are familiar with the system, which I assume is a lot of you, these are kind of the newer pieces in GNPS2 that will give you a taste for kind of the downstream or the up and coming workshops and get you a little excited about that. So any
Awesome.
questions?
I have one.
Before we move to the
Yeah.
Q&A,
Oh, yes, yes.
Ming, let's maybe just like wrap up really quick for those who are gonna leave, and also for like the people who may watch this later on YouTube. Yes, all the seminars we will upload the recording. And yeah, at that point, I just wanna first of all thank you very much, Ming, for giving this awesome overview, and definitely a lot of exciting developments. We will provide all the links that were shared today in the description later. There was a question right now, so just and then also I will post in a second the overview of the upcoming seminars and the sign-up links for the seminar series in the chat. So in case you want to check out what we're going to do in 2 weeks and so on, then feel free to check it out. And yeah, to those of you who are gonna take off, thanks for stopping by, and hope to see you all next time. And yeah, then I would say let's open this up for interactive Q&A now. So just unmute yourself or like write your questions in the chat if you have any, and I'll stop the recording so this doesn't go on YouTube.